Okay, so let's go back. We're on this one?
Yes.
Right.
No. I spoke too soon. Golgi apparatus.
Alright, let's keep on going back.
Right
Oh,
here.
you know what? I think I don't have that. Production of, you can put cell membrane. I don't know why it doesn't have that. Alright, so now we can go back to lysosome. Alright. So I talked about this yesterday, right?
Yes, you did.
Okay, and I showed you the video. Very good. Alright, so lysosomes. We talked a little bit about lysosomes, right?
No.
No, we didn't talk about it all?
No.
Okay, thank you. So a lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. Works best, these enzymes work best in the acidic environment inside the lysosome. Okay. So it says these enzymes and lysosomal membranes are made by the rough ER and transferred to the Golgi apparatus for processing. So let me explain. Let's talk about yesterday. Yesterday we said, okay, ribosome attached to the rough ER, polypeptide made, it buds off in a transport vesicle, comes here to the Golgi apparatus, where it undergoes further processing, and your finished protein buds off into another transport vesicle and gets secreted from the cell. That's pathway, I'm going to call this pathway A, because what happens is sometimes what buds off from the Golgi apparatus is actually a lysosome. So that's when it says the enzymes are made by the rough ER and transferred to the Golgi apparatus. So let's say that this polypeptide chain that's made here in the rough ER goes into the transport vesicle, comes over here to the Golgi, gets processed, and buds off. Sometimes that's not what buds off doesn't go to the cell membrane and secrete its own protein. What it does is actually stay inside the cell, and it becomes an organelle of the cell, and it's the organelle called a lysosome. So this stays in the cell and is one of the organelles. And so what it has, so what the rough ER made in this case here was a digestive enzyme. So here we have a polypeptide chain. This is a digestive enzyme that is in the lysosome, and it stays in the cell. Do you guys remember from the video on the animal cell that we watched? Now it's been a couple of days. What did the lysosome do in that video? Do you guys remember what it did? It did something inside the cell. Savannah? Yeah, absolutely. It broke down the worn out mitochondria, and so that is one of the jobs of the lysosome, is to get rid of old and or damaged organelles. So it came over here, here's the mitochondria, and it can come over here and the ends fuse with it, and the enzymes can digest the mitochondria, and we can get rid of old organelles that way. Another example of what lysosomes can do is in unicellular organisms. So this is like our cell, a little one-celled organism that lives in a pond or something like that. So here's a unicellular organism. This is only one cell big, so it for sure doesn't have a digestive system. but yet it still needs to intake its food and needs to break down its food, just like we do. So how does this unicellular organism take in their food? So here's the food particle on the outside of their membrane. So what they do is they bring in the food by taking their membrane and indenting it in and forming kind of a little bubble around that food particle. And what happens is that this membrane continues to indent and eventually pinches off, and you have this, what's called a food vacuole inside this unicellular organism. That's how it, quote unquote, eats. So now, like us, we chew up our food and swallow it, and it goes into our digestive system, but the form in which we take it in, we can't use it that way. We have to digest it. So the same thing goes with these unicellular organisms, where they have lysosomes that have enzymes in them. And so they don't have a whole digestive system, but they do have these lysosomes. These lysosomes that have digestive enzymes fuse with this membrane and dump its enzymes and mix the enzymes with the food, and then the enzymes digest the food just like our enzymes digest our food. And so that's another example of what the lysosome can do in unicellular organisms. So that's what this page is all about. So some types of cells can engulf another cell by what we call phagocytosis, and this forms a food vacuole. A lysosome fuses with the food vacuole and digests the molecules. They also use enzymes to recycle the cell's own organelles and macromolecules, a process called autophagy. In this example, you have a blank on here that is not, it says they are also involved in a cell's programmed cell death, and then there's parentheses, because that helps explain this example. The parentheses in there, it's not up here, I don't know what happened to this bullet, is called apoptosis. A-P-O-P-T-O-S-I-S. This is programmed cell death. So an example, so lysosomes help to do that. An example is the conversion of a tadpole. What was that? A-P-O-P-T-O-S-I-S. So the conversion of a tadpole to a frog. Tadpoles have tails, frogs do not. So how does it get rid of a tail? Through programmed cell death. So in those cells of the tail actually die off, and it's the lysosome and the enzymes in the lysosome that help with that. All right, we'll talk more about that later on. We have some cells during our development that also are programmed to die as well. Really young, we have tissue. Cells between our fingers and our toes, and we don't as when we're born, and so those cells are programmed to die. All right, so lysosomes play a role in that. So this picture here shows you that what I just tried to explain up here. So this is the lysosome digesting food. So this is like your unicellular organism. That's your food particle right there on the outside. The membrane indents. Just turn on one of the lights. The membrane indents, and you get your food vacuole, and the lysosome fuses with it. Enzymes mix, and you get the digestion of your food. And then the same thing with the old mitochondria in this case here, digesting it. All right, and I already showed you the animation yesterday. So just to show you how important even a small little organism like or organelle like lysosomes are, the absence of a particular lysosomal hydrolytic enzyme, that means the enzyme that breaks things down, can lead to different diseases. And one disease, very common disease, is called Tay-Sachs disease. Tay-Sachs disease, the lysosomes and cells of your brain break down lipids. And so people who have this disease, what happens is their DNA has a mutation. And remember, your DNA contains instructions to make proteins. And so what happens is, if somebody has a mutation in their DNA, and it's the DNA that gives instructions to make the enzymes and lysosomes, then what happens is that when they go to make their lysosomes, they don't have the right instructions. So therefore, they might not put the right amino acids in the right chain, which then, remember, interferes with the R groups and how they bond and give the three-dimensional shape, which then affects the function. And so somebody with this disease has this mutation in that gene. They go through this process to make their lysosomes, but they don't have a good functioning enzyme in there. That enzyme is supposed to get rid of and break down some of these excess lipids. That doesn't happen, and so what happens is they end up getting buildup of lipids in their brain, all right, and that has a lot of different side effects and so on. So that just shows you, again, to kind of pull and make connections between the nucleus and the DNA and your lysosome, because your lysosome has proteins in it that are made by your DNA. All right, so still going on with the endomembrane system here, vacuoles. So we just talked about a food vacuole. These guys are vesicles that are derived from the ER and Golgi apparatus. So this was pathway A, the protein can be secreted. Pathway B, it stays in the cell. And sometimes it becomes what builds off of the Golgi apparatus is a vacuole. And so let's look at some different kinds of vacuoles. So the food vacuole, this is in unicellular organisms formed by phagocytosis. Phagocytosis is this process here, the indentation of the membrane. There are contractile vacuoles found in many freshwater protists, which pump excess water out of the cells. And then there's central vacuoles found in many mature plant cells, hold organic compounds, pigments, poisonous compounds in water. So we talk about the contractile vacuole first. A protist is a unicellular eukaryotic organism. So eukaryotic organism means it contains what? What does it have? Nucleus and organelles, right? And it's unicellular, meaning it's only one cell big. And so a paramecium is an example of a protist. So here we have a little paramecium. They live in fresh water. And what happens is water diffuses through their membrane into the paramecium. And if water continually diffuses into the paramecium is going to swell up kind of like a water balloon and eventually burst, which is not good for the species of paramecium. So therefore, these guys have a contractile vacuole. A contractile vacuole is a sac inside the paramecium that when water diffuses in, the water accumulates in this contractile vacuole. Why it's called that is because imagine that this is a sphere, all right? So it gets bigger as it fills with water, and eventually it contracts. It squeezes like this, and all the water, it squirts the water. It's kind of like a little pump. It squirts the water back out into the outside environment. So therefore, this contractile vacuole in this paramecium is always filling up with water, contracting, squirting it out, filling it up with water, contracting it, squirting it out. So therefore, the paramecium never explodes. And so that's what a contractile vacuole is, and that's what it means by pumping excess water out of there. This next kind of vacuole is a central vacuole, which is found in, we talked about that when we looked at plant cells. When you look at a plant cell, the plant cell has a cell wall on the outside. It has a cell membrane on the inside. And then it's filled with cytoplasm, it has chloroplast and nucleus and so on. But the majority of the space within the cell is comprised of this thing called a central vacuole. And the central vacuole, as it says here, stores lots of different things. The main component is water. So like when you water a plant, the plant takes in water from the roots and it goes up the stem, and the water goes into all the plant cells and goes into the central vacuole. So the central vacuole kind of balloons up, and when it does that, it puts pressure on the outside of the cell. So the cell is very, very taut, all right, because it's filled with water. And so imagine in a plant, in a stem of a plant, you have all these different cells, and if you water your plant, it's taking in water, and all these cells, the central vacuole, are filling up. These cells are very, very straight, all right, and firm, and therefore the plant stands up straight. What happens when you don't water a plant? Well, it eventually dies, but before it dies, it'll start to wilt, right? Why is it wilting? Because the plant's using some of this water, and the water's coming out of the central vacuole, and they're using it to live, and the central vacuole is shrinking down, which means that there's not enough pressure and these cells don't have as much shape, all right? And so therefore they kind of wilt, which is why our plants wilt. Certain plants, like the petals of flowers that are like red and yellow and so on, those are plants that are also made out of plant cells. They have pigments in their central vacuole. That's the organic pigments that give them the color. So they have these pigments in the central vacuole that give them the color. Plants that are poisonous and have poisonous compounds, they're often stored in the central vacuole as well. All right, and so either poisonous because if you eat them, or sometimes they secrete poisonous things like substances like poison ivy does. All right, and you get the oily residue on you. All right, so that's your central vacuole. That's just a picture of an actual central vacuole in a plant cell. All right, so that wraps up the endomembrane system. Okay, so the endomembrane system, remember, is the nuclear envelope. It's continuous with the rough ER, which is continuous with the smooth ER. And those are actually connected together. So they're physically connected together because the membranes flow one right into the other. But we can also add in transport vesicles, the Golgi apparatus, and the plasma membrane in there. Even though the Golgi apparatus isn't physically connected to the rough ER or the smooth ER, it essentially can be connected via these transport vesicles, because the transport vesicle could butt off of one and bond and fuse with the other. All right, and so that's the whole endomembrane system. All right, and that's the end of the endomembrane system. So there's just a few other organelles that we need to just touch on to make sure that you understand. The first two are the mitochondria and the chloroplasts. These have nothing to do with any of these other organelles, so it's nothing to do with the endomembrane system. They are kind of standalone organelles. So the mitochondria is the site of cellular respiration. It's a metabolic process that uses oxygen to generate ATP. What cellular respiration does, let's just get, ATP is your main energy molecule. So when we talked about monosaccharides and glucose, you take in glucose through your diet. Glucose is the main energy molecule that we get energy from in our diet, but what we do with glucose, that glucose goes to all the bloodstream, it goes to all your cells, it goes to the mitochondria, and what we do is we break that glucose down. That releases energy. That energy makes this molecule called ATP. So this is the actual, this is cell energy molecule that's made from glucose. So that's its importance. Chloroplasts we don't have, the plants and algae have them. Those are the sites of photosynthesis, and then we'll look at peroxisomes. This is oxidative organelles. Peroxisomes break down some harmful things in the cell. So let's look at the mitochondria and chloroplasts. They actually have a lot of similarities with bacteria. Mitochondria and chloroplasts are organelles. They're not cells, but they have things in common with the prokaryotic cell. So, what are the things they have in common? It's enveloped by a double membrane, contain free ribosomes and circular DNA molecules, and grow and reproduce somewhat independently in cells. This led to a theory called the endosymbiont theory. Okay, so mitochondria and chloroplast. So here's a chloroplast, here's a mitochondria. They both have circular pieces of DNA in them, and they have ribosomes that I'll represent by dots. And so both of them have their own DNA and ribosomes. And bacteria, that's pretty much all bacteria have. They have the cytoplasm, they have their own, remember the bacteria I said has one big circular chromosome. If you were to take it out and unravel it, it would be much bigger than the bacteria cell. It has one circular chromosome or piece of DNA. So this woman, that her name was Lynn Margulis, came up with this endosymbiont theory because she thought, well, the chloroplasts and the mitochondria are kind of unique. They're the only organelles that have their own DNA, they have their own ribosomes, and in studying them, we found that the DNA that they have, the genes and these circular pieces of DNA, remember genes code for making proteins, and they have ribosomes to make their proteins, So the mitochondria actually makes its own proteins that are involved in what happens there, which is cellular respiration. So it makes its own enzymes and proteins to do its job. Chloroplasts do the same thing. The genes inside the chloroplast have instructions to make the proteins that are available, used in photosynthesis. And so they can make their own proteins there, and they don't need the cell to make their proteins to do photosynthesis. They don't need the cell for the mitochondria to make the proteins that are for cellular respiration. So her thought was that if they can do that, maybe millions of years ago, since they're so similar to our very basic cells, the prokaryotes, the bacteria, since they're so similar, maybe millions of years ago, they were actually free-living organisms on their own, instead of being a part of a eukaryotic cell. So that's what the next page here is, kind of bullets her theory. The endosymbiont theory suggests that an early ancestor of eukaryotes engulfed an oxygen-using non-photosynthetic prokaryotic cell. Oxygen-using, that's the mitochondria, non-photosynthetic, can't do photosynthesis. Prokaryotic cell doesn't have a nucleus. The engulfed cell formed a relationship with the host cell, becoming an endosymbiont. The endosymbionts evolved into mitochondria, and at least one of these cells may have taken up a photosynthetic prokaryote, which evolved into a chloroplast. I'll explain that here in a picture. So this is what kind of sums up what she thought. So on the top there is the ancestor millions of years ago, you have a eukaryotic cell. Notice that eukaryotic cell at the top doesn't have a mitochondria or a chloroplast. So then, since the mitochondria has its own DNA and ribosomes, her theory says, okay, well maybe it was its own little prokaryotic cell. So when it says engulfing of the oxygen-using non-photosynthetic prokaryote, that's what eventually we're going to call a mitochondria. So it engulfs it. See the indenting of the membrane? It's kind of engulfing it the same way that the food vacuole worked, all right? And so then it became a mitochondria and stayed and was helpful for the cell. And then she said, okay, so if that happened, maybe at least one other cell, maybe more, took in another prokaryotic cell that made enzymes to do photosynthesis, and that became the chloroplast. So that's her theory based upon the fact that mitochondria and chloroplasts have similarities to prokaryotic cells, that maybe they were outside the cell at one point. So let's look a little bit more in depth about the mitochondria. In eukaryotic cells, they have a smooth outer membrane and an inner membrane folded into cristae. The inner membrane creates two compartments. We call the inner membrane space and the mitochondrial matrix. Some metabolic steps of cellular respiration are catalyzed in the matrix. And the cristae present a large surface area for enzymes that synthesize ATP. So let me go through and explain this here. Okay, so we've talked about the mitochondria has the smooth outer membrane, and then this inner membrane has folds. The folds increase the surface area, and these folds have a special name. They're called cristae. And so inside the mitochondria, we have the matrix, and the space in between here is called the inner membrane space. And so, these folds, what this tells me is that these folds are really, really important in what occurs here. And what occurs in the mitochondria is cellular respiration. What happens and why these folds are important is that this membrane, this inner membrane, is much like the cell membrane. It has a lipid bilayer, it has proteins embedded in the membrane, and so on. And so what we have found is that in this membrane, there are a bunch of proteins embedded in the membrane that help to make ATP, which is what cellular respiration, all right? Cellular respiration makes ATP. these proteins help make ATP. So do you see if we have more surface area of the inner membrane, we have more proteins to help make ATP, which means we can make more ATP. All right, does that make sense? Okay, and so that's the importance of that. And so the surface area compared to the compared to the structure of it. And so this picture here just shows you another computer-generated picture of what I just drew on the board. So it shows all the different parts. It shows the infolding. It shows the ribosomes and the DNA and so on. All right. So that is your mitochondria. So let's look at your chloroplast. Chloroplast, you've heard of chlorophyll, yes? All right, contains the green pigment chlorophyll, as well as enzymes and other molecules that function as the key light-trapping molecules in photosynthesis. Chloroplasts are found in leaves and other plants, other green plants. And so, let's look at the structure of the chloroplast and see how it relates to its function, which is to go through photosynthesis. Photosynthesis is making what for the plant? What is photosynthesis? What are you making? Glucose, right? Using light energy as the energy source. So if we look at the structure of the chloroplast, notice here that we're showing you kind of a cross section here. There's a double membrane on the outside. The double membrane is smooth. So that tells me right there that the double membrane probably isn't involved in photosynthesis. If it was involved, there might be a higher surface area like the inner membrane of the mitochondria. So here we have this double membrane that's smooth, no folding. But what do we have a lot of inside? If we look at what we have a lot of inside, we have these discs called thylakoids. They're kind of like, look, they're stacked up kind of like poker chips, all right, or pancakes. And each one of these flattened discs is a thylakoid. The whole stack is called a granum. And we have lots of them, and this shows you a cross-section of it. When you look at the cross-section of it, it kind of looks like this. There's, each one of these is a thylakoid, the whole thing is a granum, and then there's space inside there. All right, so this is the cross-section. This line here, though, around it is the membrane, and it has a membrane just like the cell membrane. So again, it's a lipid bilayer, there are proteins embedded in the membrane, and so on. This chloroplast is the site of photosynthesis, right? So what do we need for sure for photosynthesis? We need light. And so the previous page mentioned chlorophyll. So chlorophyll is a pigment. Guess where chlorophyll is at? What do we have a lot of? We have a lot of thylakoids and a lot of membrane. The pigment is actually embedded in the membranes of the thylakoid. So do you see that the more thylakoids we have, the more pigments we have, like chlorophyll, the more light we can absorb. And if we absorb more light, what can you do more of? Photosynthesis, right? And make more glucose. So again, this shows you how the structure of the chloroplast helps with the function to do photosynthesis. You have all these flattened sacs that contain chlorophyll that help the plant to absorb the light energy to do photosynthesis. Okay? Alrighty, so that's the chloroplast. Let's just write about the structure here. So the thylakoids are those membranous sacs stacked to form a granum, and the stroma is the internal fluid inside. And the chloroplast is just one type of organelle that's under this umbrella called a plastid. That's why they're chloroplast. It's part of this plastid group. But we just need to know the chloroplast for this class. All right, peroxisomes are next. Peroxisomes are specialized metabolic compartments bound by a single membrane. So they just look like a little ball in the cell. they produce hydrogen peroxide and convert it to water. They perform reactions with many different functions. Usually they're destroying harmful molecules inside the cell. And when they do this, hydrogen peroxide is often a byproduct. If hydrogen peroxide gets inside your cells, it can be deadly, all right? And so you have these peroxisomes in your cell, and it's breaking down molecules, harmful molecules, but making hydrogen peroxide, which is H2O2, inside the cell. And if this peroxisome bursts and the hydrogen peroxide gets out here, it can cause problems. So therefore, as it makes hydrogen peroxide, it then breaks it down right away and converts it to, hydrogen peroxide can be broken down into water and oxygen gas, both of which neither one of them are harmful to the cell. And so it makes hydrogen peroxide but then breaks it down so it's not harmful to the cell. And that's the peroxide part, is where peroxisomes get their name, all right? It's the byproduct of destroying other things in the cell. All right. And this is an actual picture of the peroxisome. See, it just looks like a circle. All right. So realistically, in a cell, you just looked at a cell, we probably wouldn't be able to tell the difference between what a peroxisome looks like, what a lysosome looks like, what a transport vesicle looks like. They all look like these small little circles in the cell. All right, really. So they look like. All right, getting near the end. The cytoskeleton. It's a network of fibers extending throughout the cell cytoplasm. This one you have to put in. It helps give shape and support to the cell. So it's made out of microtubules, microfilaments, and intermediate filaments. The micro part means small. Intermediate filament means that these filaments are a little bit bigger than these filaments. All right, that's basically what that is. Filaments refers to more thread-like structure, tubules, tubes. All right. So this is the part when I showed the animal cell video. When they first entered the animal cell, the narrator was like, "Whoa, look at all the," and there was a whole bunch of like fibers and stuff all throughout the cell. That's the cytoskeleton. So realistically, when we draw cells, hardly any pictures of cells ever show the cytoskeleton. So besides all of these organelles, throughout all of this cell, you'd have all these fibers going from one part of the cell to the other, all these tubes all along, and that helps. It's kind of like the framework of the cell. So that's when it says it helps to give it shape. It's like a scaffolding system. All right, is what the cytoskeleton does. Another feature or thing that the cytoskeleton does, and this is specific to the microtubules, is it's kind of like a train track almost, or system in the cell. So this right here is a vesicle, like our transport vesicle. Transport vesicles have to go from one part of the cell, like the rough ER, to the Golgi apparatus. So how those can go is they can actually kind of ride on a microtubule. So notice here on the vesicle there's this motor protein, and the motor protein interacts with the microtubule, and so the vesicles to move, they have this motor protein that they follow along the microtubule, all right? So it's kind of like this train track system, and that's how some organelles get from one place to another, is on the microtubule there, all right? So that's that. They illustrated that in that video. I don't know if you caught it or not. All right, so that's the cytoskeleton. And then we have centrosomes and centrioles. In animal cells, microtubules grow out from a centrosome near the nucleus. This is involved in cell division. And the centrioles are within the centrosome. There's two of them. They're also made out of microtubules. So they look like this. So they're up here, it's highlighted in yellow here by the nucleus. This is the centrosome, and then the two centrioles. What that looks like, I'm not just going to draw it here. So this is a cell getting ready to undergo cell division. You have a centrosome region with two centrioles. All right, those are these two right here. So what happens is the centrioles divide and you end up with 2 of them. And then what happens is from the centrioles, which are made out of microtubules grow out from that. This is when the cell's getting ready to go through cell division, this is starting to form what we call the spindle or the spindle fiber. Does that ring a bell at all? All right, good. And so if it doesn't, let me just, in the other hours that ring a bell when I do the next picture here. These spindles help to organize the chromosomes in cell division. So eventually these centrosomes and centrioles are on opposite sides of the cell, and they look like this all across the cell like this, but they aid in the lining up of the chromosomes down the middle of the cell. Do you guys remember that from mitosis? Yeah. All right. So this is your chromosomes. All right, lining up down the middle. And so all I need you to know right now is that the centrosomes make the microtubules that help in cell division like this. Okay. Some cells also have cilia and flagella. They control the beating. Microtubules control both of these two. They're both made out of microtubules, flagella and cilia. And so they're both aiding in movement, which is what the next picture shows you here. So here's a sperm cell with 1 flagella. All right, and so it aids in the direction and movement of the sperm cell. Cilia looks different. Actually, the paramecium has cilia, so paramecium looks like this, and cilia are all these little hair-like projections. So they're short, but there are many of them, which is different than the flagella. And this shows you a picture. These are all cilia on the outside of a cell. And so the cilia move as well to help with the aid in the movement of the unicellular organism often. That's what it is. All right. Yep. All right. Last one. Get it done. And then we have the cell wall. Extracellular structure that distinguishes plant cells from animal cells. Some other types of organisms, prokaryotes, fungi, and some unicellular eukaryotes also have cell walls. It protects the plant cell, maintains its shape, and prevents excessive water uptake. We kind of already talked a little bit about the water with the central vacuole. And the plant cell walls are made out of cellulose, which is a polysaccharide. All right. Let me talk about the handout that you picked up here. I want you to do this for tomorrow. We do have homework. You're getting quite a bit of homework. Don't get out the handout sheet to write something down. Look at the picture side. So, okay, so this is the picture. What I want you to do, I'm going to have you write these 4 things down. So just write it down. Protein secreted from the cell. The next one, I just wrote protein lysosome, but protein within a lysosome. That's what I mean there. Protein secreted from the cell. Protein within a lysosome. Green protein put into a cell membrane. And a lipid secreted from the cell. What I'd like you to do is, notice I have here 4 different colors. I would like you to, on that picture in 4 different colors, to trace the pathway, where a protein that's going to be secreted from the cell, where it's going to be made, and what is the pathway it's going to take to actually be secreted from the cell. This is the pathway kind of we talked about on the board here yesterday. And so I want you to trace it, and then what I'd like you to do is to write next to it. Let me show you what I did here. So right next to it where it starts and then what organelles it will go to, as it moves through the cell. So a secreted protein starts in the rough ER, right? Then it buds off into a transport vesicle. Then it goes to the apparatus. Then it gets put into another transport vesicle. And it fuses with the cell membrane and is secreted from the cell. So you're kind of making a pathway. Alright.
So you can trace
And then the opposite side is just another couple surface area questions I want you to do on your own after we get on.
And then we can do the questions too. Is that what you just
Don't
said?
forget your phone.
Wait, on the other side? We have to do
Yeah,
the
there's a
other
couple.
side.
Yeah.